Okay, so this is investigating the effect of carbon dioxide concentration on the carbon dioxide concentration or temperature on the rate of photosynthesis. Okay, so do you think that carbon dioxide concentration or temperature are the factors required for photosynthesis?
Yes, they are.
Right? So now see, this is something fixed, and it is used for all experiments used in photosynthesis, that whenever we have to show the effect of carbon dioxide, we use a compound known as sodium hydrogen carbonate, right? And it's written in the this text box is saying, if you are going to investigate the effect of carbon dioxide concentration, you will have to use sodium hydrogen carbonate and a spatula. Of course, spatula is just to, scoop the sodium hydrogen carbonate. Now, if the examiner asks you, which he can, that why are you using sodium hydrogen carbonate? So your answer will be, it provides, or it is a source of carbon dioxide in the experimental setting, right? So I am also going to add this. Sodium hydrogen carbonate is, sodium hydrogen carbonate, it is a source of carbon dioxide in an experimental setting. Right? Because of course, I can't just grab carbon dioxide from the environment, from the air. So I have to have some artificial supply of carbon dioxide, which in this case is sodium hydrogen carbonate, right? And then it also says that we are investigating the effect of temperature. So of course, the way by means of which we can vary the temperature in any experimental setup is to use an ice bath or a hot water bath, right? And then we can keep varying the temperature by either removing some ice or adding some more ice. For example, let's say if given a choice about what kind of temperatures to choose, I would choose 0 degrees, 10 degrees, 20 degrees, 30 degrees, 40 degrees, right? So it's very easy. How will I vary all these temperatures? I will take some ice, and let's say for 0 degrees, it will be more ice. For 10 degrees, it will be less ice. And then, for 30 and 40, it will be like this. Whenever you are choosing a range of temperatures, you should always choose a range which should not be a very weird range. For example, you should not choose 0 and 13 degrees and 25 degrees. The space between the ranges should be equal. I hope you're understanding.
Yes,
right? So these are the different kinds of
like understandable. It shouldn't be just out of the world kind of thing.
Exactly. And these are like small questions which they ask. What care should you take, or how should you do that? How should you how will you change the various temperatures and so on, right? Then let's see what they are doing. Yes, we have to vary CO2 concentration also. So we can vary that by adding different masses of sodium hydrogen carbonate to the water that the plant is in. Makes sense, isn't it? For example, I'm making a sodium hydrogen carbonate solution. So let's say for, if I'm choosing 5 different carbon dioxide concentrations, maybe for the first one, I'm using 1 gram. For the second one, I'm using 2 grams and 3 grams. But then again, these concentrations also should be equally spaced. They shouldn't be weird, okay? Temperatures should also be equally placed. And this is a general rule for all experimental settings, that we use at least 5 values for our independent variable. Since the word independent variable has been used over here, so let me teach you about independent and dependent variables. First, let me check your understanding, Hamza. So you tell me, first of all, you tell me, what do you understand by the word variable?
So variable is anything that is changed or kept constant throughout the experiment. So it's like different, so it's basically it's stuff that you control or change throughout the experiment. So basically affects the experiment in a way.
Correct. Okay. Now, you have the understanding of variable. So moving on to further into the depth, which is independent variable and dependent variable. So any idea what is the independent variable?
Yeah, so the independent variable is something that you keep constant. Sorry, independent variable is, yeah, something that you change throughout the experiment to test the results. So suppose if it was for the light intensity, right? the distance from the plant itself would be the independent variable. So it's something that I can control in the experiment. And the dependent variable is directly the opposite. The dependent variable is simply the variable or something that is dependent on the independent variable. So suppose the rate at which the plant itself grows, that's going to be the dependent variable.
Correct. Very well described. I'll just add one more thing which you did not mention.
Mhm.
Yes, independent variable is the variable which we change or which we manipulate, right? And the
Mhm.
dependent variable, very easy way to remember is the dependent variable is the one which you measure.
Yeah, okay.
Because this is the easy way to remember. Yesterday, I was asking this question in my class, right? My physical class, and the students were saying, they were A-level students, and I was so shocked. They were saying, independent variable is the variable which is independent, and the dependent variable is the variable which is dependent. This does not give you any clue, right? Any clue to the understanding of these variables. So, yes, dependent is the one which you measure, and independent is the one which you change. And the independent variable is always plotted on the x-axis. Always plotted on the x-axis, and the dependent variable is always plotted on the y-axis. So I hope that's clear.
Yeah, it is.
Going back. So what is your experiment saying? It's saying you should aim to have at least 5 values of your independent variable. Think about other variables that you should try to keep constant and how you will do this. So in this scenario, we are studying the effect of varying carbon dioxide and varying temperatures on the rate of photosynthesis. So keeping them on one side, what are the other variables that you might want to keep constant in this particular setting? Yes, Hamza?
These five values of independent variables that I would want to keep constant by this experiment.
Mm.
So this is the one which the carbon dioxide, right?
And temperature, both. We are doing it together.
Okay, so maybe the amount of water given to the plant themselves, or
Okay,
perhaps the
okay,
light intensity must be kept the same.
correct. And
Those
okay,
are the
water and light intensity. I think that's more than enough. Acha, another thing, you use the word amount of water. Never now use this word, okay? You
okay.
are an O-level student. Very soon you will be appearing for AS next year.
Yeah.
So never use the word amount. Always either use the word volume or mass or concentration. If you want to say water, you can use the word volume. If you're talking about anything solid or anything which has a certain mass or a concentration, you can use concentration. So don't or use the word amount. The word amount is completely discredited in CIs. Okay, don't use this word. Clear? Hamza, I hope you can hear me.
That's clear.
Acha, okay. Now writing out your plan. Of course, this is your plan, and you could structure your plan like this, that you will first of all decide what your independent variable is, and then you will decide how to vary, right? Then you will write down the values of independent variable you will use. Let's say I have decided 5 temperatures: 0, 10, 20, 30, 40, right? Then you will also note down all the variables that you will keep constant. How are you going to make sure you're going to do that? The variable that you will measure, what is this variable? The one that we measure, what do you call it?
The independent variable. The one we
No,
measure is
no, no,
dependent.
no. Dependent. Okay, so the variable that you will measure, which is a dependent, and how are you going to do this? How will you measure the rate of photosynthesis? Are you going to count the number of bubbles produced in a certain fixed time? Because you see, if you're using a water plant, that plant, if it is photosynthesizing, okay, and you're keeping it out in the sun, if it's photosynthesizing, it will produce oxygen, because oxygen is an end product of photosynthesis, and the production of oxygen will produce bubbles. So you can calculate the number of bubbles produced per unit time.
Yeah.
ठीक है? Either you can count the number of bubbles after every 1 minute, ठीक है? Or you can say that you have decided for a fixed time, let's say 10 minutes, and you're going to count the number of bubbles that the plant actually releases in 10 minutes. It's totally up to you, right? You will take into account some safety risks and how you're going to minimize them, and an outline results chart ready to complete as you work, and so on. Acha. Let's see how this is done. So they haven't mentioned. If you could start again. They didn't mention how they're doing it.
No, I don't think
Achha.
that.
Okay, so I think they are only teaching you how to plan an experiment. Okay, fine. And that's also important because planning an experiment is a skill which many students don't know. So fine, we've done planning. Let's do this one. I think this is the last one, or maybe the second last one. Investigating the effect of light and darkness on gas exchange in an aquatic plant. Of course, the light has a certain effect on the gas exchange, and the darkness also has a certain effect. In this investigation, you will use a liquid called hydrogen carbonate indicator to look for changes in carbon dioxide concentration in the water around the aquatic plants. You will make careful observations of color changes and then use your results. Okay, now let me tell you another thing. In the previous experiment, Hamza, when we wanted to study the effect of varying the carbon dioxide concentration, we used sodium hydrogen carbonate as a source of carbon dioxide, right?
Yeah.
But over here, there is another compound whose name resembles the name of that compound which we used in the previous experiment. That compound's name was sodium hydrogen carbonate. This is known as hydrogen carbonate indicator. Most of the students mix up the sodium hydrogen carbonate and the hydrogen carbonate indicator. What are their differences? The differences are that the sodium hydrogen carbonate is used as a source of carbon dioxide, whereas the hydrogen carbonate indicator is used as an indicator to check the acidity or alkalinity of a solution. In other words, to check the pH, right? Tell me, if the plant leaf is exposed to light, it's there are two leaves. This leaf is exposed to light and this leaf is exposed to dark. What do you expect? Which gases will move in and out of the leaf which is exposed to light?
Carbon dioxide will move in and oxygen will move outside.
And what about this one?
There will be no displacement in the gases, right? Because no photosynthesis no photosynthesis is taking place, so the plant won't really need carbon dioxide. So only O2 will
But
go
will
in.
but then will the plant respire? You can't stop the plant from respiring.
Okay, yeah. Oh, okay, I get it now.
Got
Wait,
it?
wait.
So
Okay,
now,
so in
yeah,
photosynthesis, right? So photosynthesis
huh?
it's is the so hello? Yeah.
Yes, better. Repeat what you said.
Yeah, I was just saying photosynthesis itself is the production of glucose, right? But photosynthesis doesn't provide
Yes,
energy. So to produce energy, a plant needs to go through aerobic respiration.
of course,
Okay,
of
and for
course.
that means that the plant can take in CO2 for photosynthesis, but at the same time, it's taking in O2, oxygen, for aerobic respiration.
Correct.
So then, Miss, wouldn't it
Right?
be better for yeah, so wouldn't it be better then for the plant to produce so you know how in photosynthesis the final product is glucose plus oxygen? So why doesn't the
Mhm.
plant just transmit the oxygen that's being produced from photosynthesis to the oxygen that's required for aerobic respiration?
Why doesn't the plant transmit the oxygen which is required for, which is produced as a result of photosynthesis to be used for respiration?
Yes.
This is what you're saying.
Yes.
See, oxygen will come out through the stomata, right? And it will be released by diffusion into the atmosphere.
Okay.
The plant can't do anything about it. And since the plant is, of course, releasing oxygen into the atmosphere, it can always get back that oxygen. What will stop the plant from getting back that oxygen?
I mean, wouldn't
Oxygen
it be
can
just
come
more
inside.
work?
Huh?
Wouldn't it be just more work?
More work. Now, see,
Yeah.
it's like this. You're right. You're right. More work. In any case, it's work. Uh, if it's utilizing the oxygen from the environment or if it's utilizing its own entrapped oxygen, it's one and the same thing. It still has to do a lot of work, Respiration has to continue. In fact, you know what? Even when the plant is exposed to daytime light, which is in this scenario, don't you think the plant is respiring during this time, during the daytime?
It is, obviously.
It is, it is. But then what happens is we teach the O-level students like this. We ask them, when does a plant respire? So the first answer that comes out is they will say at night. That's the wrong answer. You will say the plant respires all the time, number one. Number two, but the rate of photosynthesis is so overwhelming. It's so overwhelming that the rate of respiration becomes very minimal. Like we can't see the effects of cellular respiration during daytime as much as we see the effects of cellular respiration during nighttime. So during daytime, photosynthesis is extremely overwhelming, right? And of course, the plant has oxygen inside. It always has oxygen inside. The oxygen, don't think that the oxygen cannot come inside. It can come inside the plant during daytime also. So it's like both the processes are taking place simultaneously, basically. It's not a wastage of energy.
Okay.
Right?
So
Acha,
you're saying
so
that the energy is produced from photosynthesis at a much higher level than what the plant is able
exactly.
to respire at. So it doesn't really matter. Because
Hmm.
eventually they do what do you call store in the form of glucose, right?
Exactly. Exactly.
Okay,
Acha.
that makes
Yes. What do we do in this case? Acha, in every situation, you must always notice, and sometimes the examiner can ask you, why are we choosing an aquatic plant? The reason for choosing an aquatic plant is that in order to calculate the rate of photosynthesis, we have to use an aquatic plant so that we can see the bubbles of oxygen emerging from that, right? For example, let's go back to this diagram. If I'm not using an aquatic plant, where will the oxygen bubbles go? They will go into the atmosphere. Okay? So how am I going to capture all those oxygen bubbles? I can't capture them. That's why I use an aquatic plant. Look, this is an aquatic plant. I want to measure the rate of photosynthesis, which in this case, I was measuring the effect of light intensity on the rate of photosynthesis. So I take an underwater plant so that I can actually monitor or measure the rate of photosynthesis by counting the number of bubbles which are produced during a fixed period of time. Is that making sense?
Yes, miss, it is. So it's simply to
Hmm?
count the number of bubbles that are being made.
Because I have to have a tangible, way of calculating the rate of photosynthesis. I can't just capture the oxygen molecules that have been released into the atmosphere. Okay. Again, we are using an aquatic plant. Let's see what we are doing. Test tubes and all fine. Achha, now, some preliminary information is given about the hydrogen carbonate indicator, which is that it's an indicator, okay? It's not the sodium hydrogen carbonate, which we used in the previous experiment. It's a totally different thing. Don't mix up this with sodium hydrogen carbonate. Okay, let's see how this indicator works. So if you use the indicator, it will change the color according to how much carbon dioxide there is. The colors are, the indicator will change to purple color when there is no carbon dioxide. Okay? It will change to red color when there is little carbon dioxide, as we have in the air, which is around 0.03%. And it will change to yellow color when there is a lot of carbon dioxide. So this means that, in other words, the indicator changes to yellow when the pH is acidic. The indicator changes to red when it is slightly acidic, and when there is no carbon dioxide, it remains purple. Right? Let's see how this is done. This is an important experiment. Pour hydrogen carbonate indicator into each of the 4 test tubes that are shown in the diagram to the same depth in each one. About a third full works well. Record the color of the indicator in each tube, right? When I say pour hydrogen carbonate indicator into each of the 4 tubes to the same depth in each one, am I controlling the volume of the hydrogen carbonate indicator? Yes, Hamza?
Yes, you are.
So am I controlling this variable? Right? Then after filling it up, you will record the color of the indicator in each test tube, the basic color, the preliminary color. Place a piece of aquatic plant in the indicator in 2 tubes. Try to use similar sizes of plant each, similar size. Again, I'm controlling the variable. I'm just putting a star wherever we are controlling variables. Use a black paper to wrap around one of the test tubes with a plant in it and one without. Okay, so we have 4 test tubes. For example, test tube, this test tube has an aquatic plant and it is uncovered with black paper. This test tube has no aquatic plant and it is uncovered with black paper. Out of these, both these test tubes are covered with black paper. One of them has the aquatic plant and the other doesn't have the aquatic plant. Now, Hamza, a task for you. Tell me, out of these 4 test tubes, which 2 are controlled?
Which two are controlled?
Yeah.
The black ones, right?
No, not the
With
black ones.
airtight rubber band. Oh, maybe the one on the, what do you call, right? Because the amount of hydrogen carbonate solution present inside of them is equalized or equal to one another.
There are 2 test tubes which are controlled over here. One is this one, and the other is also the one which is covered with a black foil and which doesn't have a plant. No plant, no plant. You know why? Because if there's no plant, will it photosynthesize?
No, it won't.
Right? So that's why these are the 2 controls, okay? And then we are using hydrogen carbonate indicator to, acha. Then stand all 4 tubes in the light and leave them for at least an hour, preferably longer. The longer, the better. When the indicator has changed color in at least one of the test tubes without a covering, remove the black paper from the 2 covered tubes, record the color of the indicator in each tube. Now see, we will expect, of course, when sunlight is falling onto this test tube, we will expect that the color will change. This doesn't have a plant, but we really don't know if the sunlight falls onto this test tube, how will it affect the color of the indicator? We'll see that. Over here, the sunlight will be blocked by the paper, and over here also the sunlight will be blocked by the paper. So these 2 test tubes, I think they should give the same color, and let's see what color these test tubes give us. So using the information about the color of the hydrogen carbonate indicator, use the information about the color of the hydrogen carbonate indicator to compare the concentration of CO2 in 4 tubes. Plants, like all living organisms, they respire. They do this all the time. Respiration uses oxygen and produces carbon dioxide. In bright light, the plants photosynthesize as well as respire. They photosynthesize faster than they respire. Use this information to explain your results. So let's go back. Okay, let me just. In this test tube, will photosynthesis happen?
Sorry, miss?
In this test tube, will photosynthesis happen? Yes or
Yes,
no?
yes, it will.
Okay. Will respiration happen?
Yes, I believe so.
Yes.
So
In
hydrogen
this,
indicator solution just absorbs carbon dioxide, right?
yes.
Okay.
In this test tube, will anything happen in this one?
No.
Nothing will happen. So no change. Acha, let's say that this had a plant and this had no plant, let's suppose. So in this test tube, will respiration happen? Yes. Will it happen?
No.
Why not? Do you need
Because
sunlight to respire?
yes, you do. Oh, respiration will happen. Yes, miss.
Will photosynthesis happen? Yes.
Photosynthesis,
No?
no.
No. Acha. Don't you think, what do you think should be the color of the indicator in this test tube?
Color of the indicator
Will
and
we
what test?
in okay, let me number 1, 2, 3, 4. Color of the indicator in test tube number 1.
Uh, pink, I guess.
Let's go back and see.
Oh, okay.
Pink.
Purple. No. Purple, red, yellow. So it needs to be no carbon dioxide. It'll be yellow, right?
Do you think that there's going to be a lot of carbon dioxide? Acha, the test tube, okay, let me draw this test tube. This is the test tube. There is the
Okay.
plant. Okay, sunlight is coming. So this photosynthesis is happening more than respiration. Okay, it's happening more than respiration. So what do you expect in this scenario? Will
Photosynthesis is
there
happening
be,
within respiration, so
huh,
there will be carbon dioxide present, miss.
there'll be more carbon dioxide. There'll be more
Yeah.
carbon dioxide present. When photosynthesis is happening, the carbon dioxide gets into the leaf.
Oh, okay, that makes it okay. So purple.
Purple. So do you think there's no carbon dioxide in this? No. The plant is respiring also.
Oh, sorry, it's red.
Now you
Uh,
get it?
yes,
These
now I
kind of
get.
questions are not that easy. Okay. So let's go to the, okay, let's write about this test tube number 1. So the color is red. Okay. And the reason is, you will write down, photosynthesis is high. So CO2 is used and respiration produces CO2. This much to give a red color. Now, did you get it?
Yes, miss.
Okay. Moving on to test tube number 2. In test tube number 2, will you see any change? Test tube number 2, I think it should be purple, isn't it? Yes, Hamza?
Yes, miss. It should be purple. In
It
test
should be
tube number
purple,
two,
right?
yeah.
It should be purple because actually, if you look at this test tube, it has a very tight rubber bung. So this rubber bung shows that air can't go inside. If the air is going inside, then it might change a little bit red color, but it's purple, right? Moving on to plant 3. Plant 3 has a plant, but it's covered by sunlight in which respiration happens, but the photosynthesis doesn't happen. So, what should be the color? Should the color be yellow? Lot of respiration. Yes. Let's look
Yes,
at this.
miss.
Yellow
Yellow.
when there is a lot of carbon dioxide, right? So this test tube, which is test tube number 3, which has a plant which is covered by black paper, it can't absorb light, so it doesn't photosynthesize. Only respiration is happening. So this test tube will be yellow in color. Yellow. Increased CO2. And test tube number 4, color.
Test tube number 4 is color. There's nothing, right? So there will be carbon
Nothing.
dioxide present inside,
Purple.
right, miss? Yeah, so it'll be
Purple.
purple.
Purple. Okay. Acha.
Yes.
Now I'm ending my meeting and restarting it. This is also one of the disadvantages of not joining that platform because you have to end it and then rejoin it. That is an unlimited because that's a paid account, LMS, right?
Yeah, okay.
Okay. So I'm ending and then we'll restart.